All right, so we are here for our last quick little video for this week's learning targets, and it is all about other fuel sources and regulation because any chemical reaction needs to be able to be regulated. So the first thing we're going to talk about is how different foods are broken down in respiration, because we talk about this when we talk about respiration. We start with glycolysis. We always start with glucose. I don't know about you, but I don't run around eating sugar all the time. I mean, it tastes good, but or carbs. I do eat a lot of carbs, but that's not all I eat. I eat protein, I eat fats. And so where do those come in? So this is a really good graphic for that. I don't know if it's going to let me. Oh, there it goes. So let me present. Um, but what you can see here is that, yes, those sugars get broken down into monosaccharides, which are going to enter, for the most part, at the beginning of glycolysis. There are some sugars that enter, I think maybe fructose. I'm not sure if that's 100%, but I'm pretty sure fructose enters a little bit lower in the steps. So that's what this arrow is showing, some different sugars that come in maybe 2 or 3 steps, because glycolysis is actually a 10-step process. So glucose comes in on step 1. Some of these other sugars might come in step 2 or 3. What if you eat protein? What if you eat a hamburger and you got that meat in there? Well, you're going to break that down into amino acids through digestion. And then those amino acids are going to be converted to pyruvate or acetyl-CoA or some other part of the citric acid cycle. So they're going to enter a lot lower. Well, if you'll remember, we make 2 ATP in glycolysis. That means you're not going to make those 2 ATP because it's coming in at the bottom of glycolysis. So those kinds of things are going to impact your numbers of ATP produced. Fats, you break down into glycerol, which you would convert to glyceraldehyde 3-phosphate, which is about, I think, about halfway through, maybe it's near the end of glycolysis. But anyway, so it's going to come in midway through glycolysis, and then your fatty acids that are attached to that glycerol are broken free and converted to acetyl-CoA. So they're going to come in even lower. So again, they're not going to make those 2 ATP that glycolysis makes. So you're going to see a little bit less ATP when that happens. So you are able to use other fuel sources besides the carbohydrates. They're just going to enter the process at different places. All right, the other thing that I wanted to talk about really quickly, let me use good English, is regulation. And so there was one example in your PowerPoint that talked about phosphofructokinase. ACE tells you it's an enzyme. And what we always want to do when you look at metabolic processes, and I included these pictures, but I didn't want them to scare you. This is glycolysis. So like I said, it's a 10-step process, and every single step has an enzyme. So you see hexokinase, you see phosphofructokinase, which is the one on the other slide. If you look at citric acid cycle, it's an 8-step process. Each of those steps is going to have enzymes that impact that step and help that step occur. We need to be able to regulate those enzymes because we don't want to do this process if we don't have any acetyl-CoA. We don't want to do this process if we don't have any glucose. That would be a waste of energy, and it would just be spinning your wheels because there's literally no fuel going in. So what we do instead is we are able to regulate them. And so there are negative and positive feedbacks. Negative feedbacks are usually where some product is going to go back and negatively turn off that enzyme. So phosphofructokinase, which if you'll remember was up here in step 3, is negatively regulated by citrate and by ATP, because if you're making plenty of citrate, that means you've got plenty of pyruvate, plenty of acetyl-CoA. Okay, so that citrate is a product of acetyl-CoA and that 4-carbon. So when you have lots of citrate, it'll tell phosphofructokinase to just chill for a minute. Like, we're good. We have plenty of citrate, which means we can do plenty of citric acid cycle and plenty of oxphos. If you have a ton of ATP, same thing. If we've got a lot of ATP, then we can just take a break. We can rest a little bit. And so your body does that to save energy. You don't need to do these processes, even though they're producing ATP. Their energy burning, I guess you could say. They're using energy, and so you don't want to do them if you don't have to. Positively regulated, AMP, which is what we get when ATP gets broken all the way down from ATP to ADP to AMP, that means you're starving. Like, I need some energy. And so that's going to tell phosphofructokinase to kick it in high gear because we're running out of energy. So I can explain why it's regulated, because you don't want processes to occur that don't need to, because that's a waste of energy, and things that waste energy die. And then I can explain how it's regulated. Again, usually products or precursors are going to impact different enzymes, and all enzymes are able to be regulated. I hope that helps. And if you have any questions, let me know.